Welcome to Bedtime Astronomy. Explore the wonders of the cosmos with our soothing Bedtime Astronomy podcast. Each episode offers a gentle journey through the stars, planets, and beyond, perfect for unwinding after a long day. Let's travel through the mysteries of the universe as you drift off into a peaceful slumber under the night sky.
I want you to put yourself in a highly specific, highly stressful scenario for a minute.
Okay, let's hear it.
So you're standing inside a pressurized habitat module, and you are on the 4th human mission to Mars.
Wow. No pressure.
Right. Literally and figuratively. The air you're breathing is scrubbed, it's recycled, and it tastes faintly of like ozone and metal. And the low, constant hum of the life support system is this vibration you just feel deep in your boots 24 hours a day.
It's just the background noise of keeping you alive.
Exactly. Outside the reinforced quartz glass, it's just this freezing, irradiated, rust-colored wasteland. But your focus isn't out there on the horizon. Your focus is on what is sitting right in front of you on a metal table.
Which is what?
You are looking at a pile of dead, toxic Martian dirt. And it is your job, your specific make-or-break mandate to establish the first self-sustaining food crop for this entire settlement.
That's a terrifying amount of responsibility.
I mean, if you fail, the colony's food supply eventually just runs out. The resupply ships from Earth are tens of millions of miles away. They're locked in these orbital transit windows that are months or even years from opening. So you are quite literally holding the future of a permanent human presence in your hands. And your only raw material is a pile of poison dust.
It is the ultimate high-stakes agricultural puzzle. I mean, we tend to focus so much of our collective imagination on the rocketry.
Well, for sure, the massive orbital boosters.
Right, the sheer thermodynamic violence required to push mass out of Earth's gravity well. But those machines, they only serve one function, which is getting us across the void.
Which is hard enough.
It is. But the far more complex, far more daunting barrier to humanity actually becoming a multiplanetary species is what happens after the engines shut off.
Right.
That pile of dirt you just described represents a logistical nightmare that cannot be solved with, better thrusters or stronger steel. It requires a fundamental mastery of invisible, microscopic ecosystems.
Because if we can't force that dead dirt to sustain life, we aren't colonizing anything.
Exactly. We are just temporary visitors on a staggeringly expensive camping trip.
And that biological puzzle is the entire mission of our conversation today. We are looking at the absolute bleeding edge of bioengineering and space agriculture.
It's such an exciting field right now.
It really is. We're going to explore how researchers are planning to deploy highly specific microscopic organisms, specifically fungi, to transform completely inhospitable space dirt into lush, biologically active, life-giving soil.
Which, on its face, sounds like alchemy.
Totally sound like magic, but when you break down the mechanisms, it is pure, rigorous, evolutionary biology.
Well, to even begin to understand how these organisms perform this transformation, we really have to establish just how hostile the baseline environment is out there.
Right, because it's not like Earth.
Not at all. The ground we walk on here on Earth is fundamentally, chemically, and structurally different from the surface of the Moon or Mars. We casually use words like dirt or soil interchangeably.
Sure, I do it all the time.
But in planetary science, there is a massive distinction between soil and what covers those alien worlds, which is called regolith.
Okay, let's unpack the chemistry and the structure of that dead dirt problem. Because, the common assumption is that dirt is just a passive medium, right?
Right, like it's just a placeholder.
Yeah. You go into a backyard, you dig a hole, you drop a seed in, pour some tap water on it, and the seed does all the work. It feels like the dirt is just a cup holding the plant upright. But Earth's soil is actually this massive biological engine, isn't it?
Oh, it is a profoundly complex living matrix. A single handful of healthy Earth soil. It contains billions of organisms.
Billions. In one handful.
Billions. You have nematodes, protozoa, these sprawling networks of mycorrhizal fungi, bacteria, decomposing organic matter, worm castings, decaying leaves. All of this.
And all that organic material creates a dynamic chemical environment.
Exactly. The soil breathes. It holds water through complex capillary action. It regulates temperature. It actively cycles carbon, nitrogen, and phosphorus. The biological matter creates a physical architecture, like pores and aggregates, that allows a seed to thrive.
And regolith just doesn't have that.
Regolith possesses none of that architectural or biological legacy whatsoever.
Because it hasn't had the benefit of billions of years of life living and dying on top of it. I mean, regolith is essentially just crushed inorganic rock.
Yes.
It's basalt and silicate that has been pulverized by a relentless bombardment of meteorites over eons and then sterilized by unfiltered solar radiation.
A very violent origin story.
And it's not just that it's empty of the vital nutrients crops need, like bioavailable nitrogen, potassium, and phosphorus. It's that the regolith actively subjects any potential biological life to severe abiotic stress.
Yeah, let's talk about abiotic stress for a second. Because when we use that term in this context, we are looking at an environment that is aggressively anti-life.
Meaning non-living stress, right?
Right. On Earth, a plant might face biotic stress, like aphids attacking its leaves or some kind of fungal blight. Abiotic stress on Mars or the Moon means the environment itself is the hostile actor.
The planet itself is trying to kill the plant.
Precisely. You are dealing with wild temperature fluctuations that can literally shatter cellular walls. You are dealing with ionizing radiation that directly shears and mutates DNA. And crucially, you're dealing with a heavy, localized presence of toxic chemical compounds right there in the dust.
The perchlorates always come to mind here.
Oh, yeah. The perchlorates are a huge issue.
When the Phoenix Mars lander and then later the Curiosity rover, when they started sampling Martian regolith and finding calcium and magnesium perchlorates, it was just a massive blow to the idea of easy Martian farming.
It really dampened the mood for space agriculturalists.
Because perchlorates are highly reactive chemicals. I mean, in humans, they severely disrupt thyroid function.
Right.
And in plants, they essentially bleach and destroy cellular tissue.
They wipe them out.
So planting a seed in Martian regolith is like trying to grow a fragile tomato embryo in a mixture of crushed glass and battery acid.
That is exactly what it's like.
You have these sharp, sterile, hydrophobic rocks mixed with aggressive, toxic salts.
The crushed glass and battery acid analogy actually captures the physical and chemical reality perfectly.
Really?
Yeah, because if you look at the physical structure of the regolith, the crushed glass part, it means there is no capillary action to hold water.
So it just runs off.
If you pour water on it, it either immediately sublimates in the low atmospheric pressure or it pools in these useless toxic concentrations. There's no biological scaffolding. There's no organic carbon to feed microbial life.
Right.
And then the battery acid part, the perchlorates and heavy metals, that creates an electrochemical environment that actively dismantles complex organic molecules. It's just a sterile chemical wasteland.
Which naturally leads me to the brute force engineering question.
Okay, lead on me.
I look at this problem and I think about massive industrial chemical plants on Earth. We know how to synthesize liquid nitrogen. We know how to mine and refine phosphorus and potassium.
We do it every day.
Right. So if the Martian regolith is just empty and toxic, why do we need biology to fix a chemistry problem?
That's a fair question.
Why don't we just synthesize massive vats of liquid NPK fertilizer, ship it up to the habitat, wash the perchlorates out of the dust, and just pump the regolith full of synthetic nutrients until it acts like soil?
Well, the primary barrier to that approach is structural, but it's also deeply tied to the physics of space travel. Let's look at the structural side first.
Okay.
We touched on how Earth soil has an architecture. It forms aggregates that hold onto nutrients and then release them slowly to the plant's roots. This is largely due to something called cation exchange capacity, or CEC.
Cation exchange capacity.
Right. The organic matter in clay and healthy soil have a negative electrical charge, which actually holds on to positively charged nutrients like calcium, magnesium, and potassium.
Like a magnet.
Yeah. Exactly like a magnet.
Yeah.
Martian regolith lacks this organic matter entirely. It has an incredibly poor cation exchange capacity.
Oh, I see. So even if you saturate it with a highly refined liquid fertilizer, the regolith has no mechanism to actually hold on to those nutrients.
None whatsoever. The synthetic chemicals would simply wash straight through the crushed rock, or they would bind irregularly and create these localized zones of extreme chemical burn that would kill the plant roots anyway.
Wow.
You can't just pour complex chemical nutrients onto pulverized basalt and expect it to suddenly behave like a biological sponge. The physical and chemical nature of the ground itself must be fundamentally altered. The substrate has to be terraformed at the micro level before a plant can even attempt to interface with it.
We need something that actually builds the sponge from the inside out.
Exactly.
We need a mechanism that alters the structural reality of the dirt rather than just painting over it with fertilizer.
Which brings us to the microscopic heroes of this whole endeavor.
Yes.
Researchers are looking to highly specialized biological agents to perform this structural and chemical overhaul. They are specifically focusing on beneficial fungi to convert this toxic, nutrient-absent regolith into a hospitable matrix.
And who are the main players here?
The heavy hitters in this research are arbuscular mycorrhizal fungi, which we usually just abbreviate as AMF. They are members of the Glomeromycota phylum and also species within the Trichoderma genus.
The biology here is absolutely wild to me because, when most people hear the word fungi, they immediately picture a mushroom popping up on a damp log in the forest.
Wait, like a little toadstool.
Yeah, but we aren't talking about cultivating a crop of portobellos on Mars. We are talking about microscopic subterranean networks of mycelium.
Yes, the invisible network.
And the way these specific fungi, particularly the Glomeromycota, operate is fascinating. They are obligate biotrophs.
Meaning they cannot survive in nature without a host plant.
Right. When you introduce arbuscular mycorrhizal fungi into a substrate with a plant, the fungi literally penetrate the cellular walls of the plant's roots.
They push right through the outer root tissue and form these highly branched, microscopic, tree-like structures inside the plant's own cells.
It's like an invasion, but a good one.
Exactly. Those structures are called arbuscules, and this is the site of resource exchange. From those arbuscules, the fungal network, the hyphae, branches out into the surrounding soil, or in this case, the regolith.
And those hyphae are vastly thinner and more pervasive than the plant's own root hairs could ever be, right?
Oh, significantly.
Yeah.
They act as a microscopic biological extension of the root system.
They can weave themselves into the microscopic crevices of the crushed silicate rocks where plant roots are just completely excluded. They can't fit.
Right, the roots are too blunt.
But it's not just a matter of physical reach, is it? It's what they are doing biochemically when they get into those crevices.
This is where the magic happens.
Because a plant root sitting in Martian dirt might desperately need phosphorus or iron, but those elements are locked away inside the inorganic crystalline structure of the basalt. The plant has no mechanism to access it.
It's starvation surrounded by food.
Yes, but the AMF network does have a mechanism. These fungi excrete organic acids, things like oxalic acid and citric acid.
And those organic acids are the master keys to the rock. When the fungal hyphae excrete oxalic acid onto a piece of inorganic regolith, the acid actively dissolves the mineral structure.
Literally melting the rock.
Literally. It engages in a process called chelation, where the organic acid molecules physically grab onto inorganic metal ions, like iron, zinc, or those locked-up phosphates, and pull them out of the rock matrix.
That's incredible.
The fungi metabolize these trapped, inaccessible minerals, convert them into a soluble, bioavailable format, and then transport them through their mycelial network directly back into the plant's root cells.
Okay, here's where it gets really interesting for me. It functions exactly like a universal power adapter mixed with a microscopic mining operation.
I love that analogy.
Thanks. The plant is plugged into the Martian dirt, but the raw inorganic minerals are the wrong voltage. The plant can't read them, it can't process them.
So the fungus acts as the adapter.
Yes. It mines the rock, translates the raw inorganic chemistry into a biological format, and mainlines it directly into the plant. And in return, the plant, which obviously has access to sunlight above the surface, uses photosynthesis to synthesize carbon-rich sugars, which it then pumps down into the roots to feed the fungus.
It is a flawless, self-sustaining economic exchange. The plant trades carbon for minerals.
It's beautiful.
And looking at this from a planetary history perspective reveals a really profound evolutionary echo. Because this exact symbiosis is not some novel concept engineered in a modern lab.
No, it's ancient.
This is the oldest trick in the terrestrial playbook. The Glomeromycota phylum is ancient. Evolutionary biologists have found fossil evidence suggesting that roughly 450 million years ago, during the Ordovician period, when the Earth's surface was just barren, sterile rock, the very first aquatic plants began attempting to colonize the land.
But those early plants didn't even have root systems yet.
Exactly.
They had no way to extract nutrients from the bare rock of early Earth.
So they relied entirely on ancestral fungal networks. Fungi had already begun to colonize the terrestrial surface, mining the rocks through the exact acid exudation mechanisms we just discussed.
So they teamed up.
The aquatic plants partnered with these fungi, using them as surrogate root systems. That ancient biological partnership is literally what terraformed the Earth. It broke down the primordial rocks, mobilized the first terrestrial carbon cycles, and created the very first soils.
That is just an incredible realization. The exact same biochemical mechanism that allowed life to conquer the sterile, irradiated rock of early Earth, 500 million years ago, is the exact same mechanism we are deploying to conquer the sterile, irradiated rock of Mars today.
It's poetic, really.
We are essentially rebooting the Earth's original terraforming software on a new server.
The biological continuity is striking. When you visualize this process happening inside a Martian habitat, you are looking at a microscopic fungal superhighway.
A superhighway of life.
A vast, intricate web of mycelium actively dissolving the toxic dirt, synthesizing bioavailable nutrients, and delivering them to struggling plant roots, completely bypassing the limitations of the regolith itself.
Wait, but let's look at the limitations of the fungi for a second, because this raises a significant biological question for me.
Okay, what's the hang-up?
We established that the Martian regolith is loaded with perchlorates, heavy metals, and zero organic matter. The goal of introducing fungi like AMF and trichoderma is to relieve those abiotic stresses and create what space agriculturalists call an engineered microbiome.
Right.
But if this crushed glass and battery acid environment is too toxic and hostile for the rugged cellular walls of a plant root, why doesn't it instantly annihilate the fragile microscopic threads of the fungi?
That's a very logical question.
It seems counterintuitive that the microscopic organism is the one taking the vanguard position against the toxins.
It does seem contradictory if you view fungi as delicate. But fungi are not delicate.
They're not.
No, they are biochemical powerhouses. They possess a level of metabolic plasticity and environmental hardiness that plants simply never evolved.
Really?
Yes. Plants are autotrophs. They make their own food via sunlight, so they generally require stable, hospitable environments to operate their photosynthetic machinery. Fungi are heterotrophs that evolved specifically to be nature's ultimate recyclers, decomposers, and remediators. They're built to interface with decay and toxicity.
So how do they physically survive the heavy metals and the perchlorates that would otherwise bleach a plant cell?
They utilize several advanced cellular defense mechanisms. When a fungal hypha encounters a highly toxic concentration of heavy metals, it doesn't just absorb it and die. Many fungi produce specialized proteins called metallothioneins.
Metallothioneins.
Right. These proteins have a high affinity for heavy metal ions. They bind to the toxic metals, essentially locking them into inert, harmless complexes outside the cell membrane.
Oh, wow. So they neutralize the threat before it even crosses the cellular boundary.
Exactly. And if the toxins do breach the cellular wall, the fungi have another layer of defense. They can sequester these toxic ions into specialized internal compartments called vacuoles.
Like a little panic room for poison?
Pretty much. They essentially swallow the poison, lock it inside a microscopic vault within their own bodies, and keep it away from their vital metabolic processes. The fungi act as a biological toxin sponge. They absorb the environmental hazards, neutralizing the immediate area around their network.
That is wild. And species like trichoderma are particularly aggressive in this regard, right?
Very aggressive.
While AMF acts as the nutrient delivery network directly bound to the plant, trichoderma acts almost like a microscopic immune system for the soil itself. It actively breaks down complex, hostile compounds and outcompetes pathogenic microbes.
So these fungi aren't just surviving in this hostile environment, they are actively gentrifying it.
Cleaning up the neighborhood.
They are sequestering the heavy metals, breaking down the toxic salts, and paving the way for the plants. They're executing the engineered microbiome strategy perfectly.
And their impact isn't just chemical, it's structural too.
Yes, very much so. As these fungal networks push through the pulverized dust of the regolith, they excrete a glycoprotein called glomalin.
Glomalin.
Glomalin is fascinating. It acts like a biological mortar or soil glue. It binds the microscopic particles of sterile silicate dust together into larger aggregates.
Ah, so that's how they build the sponge.
No.
The glomalin clumps the dust together, and suddenly you have microscopic pores, you have architecture.
You introduce empty space into the crushed rock. Those pores allow oxygen to circulate, and they allow water to be held in suspension rather than sublimating or washing away.
So the fungi are structurally engineering a hospitable microenvironment.
Exactly. If you set up a time-lapse camera inside one of these sterile metallic Martian greenhouses, the transformation would be stark. You begin with this dead, rusty, lifeless dust. But as the engineered fungal microbiome takes hold, excreting organic acids and glomalin, that dust undergoes a complete metamorphosis.
It literally changes color, right?
Yeah. It darkens in color as organic carbon builds up. It clumps together. It becomes rich, moisture-retaining, fungal-laced soil.
It's a brilliant biological solution, but, it forces us to address the massive, multi-trillion-dollar macroeconomic question hanging over this entire endeavor.
The money question.
The money question. We are investing vast amounts of time, money, and research to genetically profile microbiomes, isolate specific glomeromycota strains, and test fungal resilience in low-gravity environments. We are doing all of this just to figure out how to turn toxic space rock into dirt.
It sounds a bit crazy when you say it like that.
Right. A skeptic would look at this and ask, well, if we possess massive orbital rockets, vehicles capable of carrying heavy habitat modules, automated rovers, and dense life support systems, why not just load a cargo bay full of premium, nutrient-rich Earth potting soil?
The brute force cargo method.
Yeah, why are we fighting a war against toxic Martian regolith when we could just bring the good stuff with us?
It is a question that cuts straight to the core of orbital mechanics and the physics of space travel. The simplest answer is that bringing soil from Earth is an astronomical, financial, and logistical impossibility, governed entirely by the tyranny of the rocket equation.
So what does this all mean? Let's walk through the math of that tyranny for the listener.
Sure.
The rocket equation, formulated by Konstantin Tsiolkovsky, basically states that every single ounce of mass you want to lift out of Earth's massive gravity requires a proportional amount of fuel.
A lot of fuel.
But then you need additional fuel to lift the weight of that initial fuel. And then you need more fuel to lift that fuel. It creates this punishing exponential curve.
It's a vicious cycle.
This is why a massive rocket like the Saturn V or modern heavy lift vehicles are, 85 to 90% propellant by mass? The actual payload, the thing you are trying to put into space, is a tiny fraction of the vehicle's total weight.
The margins are razor thin. When mission planners are designing a payload, every single gram is scrutinized, debated, and shaved down. Soil, healthy, living, moisture-rich Earth soil, is incredibly dense and heavy.
It's basically mud.
Exactly. Water alone weighs roughly 8 pounds per gallon.
Yeah.
Soil retains a massive amount of water weight. To pack enough Earth soil to establish a farming footprint large enough to sustain even a small crew of colonists indefinitely, you would be talking about thousands upon thousands of pounds of sheer dirt.
You would need entire fleets of heavy lift rockets dedicated to nothing but transporting wet dirt across millions of miles of space. The launch costs alone would rapidly escalate into the trillions of dollars.
It bankrupts the mission before it even leaves the launch pad. Mass is the ultimate enemy of space exploration. You cannot negotiate with gravity.
No matter how rich you are.
Right. This undeniable constraint is why space agencies operate under a paradigm called ISRU, in situ resource utilization.
ISRU, living off the land.
ISRU mandates that for any long-duration, deep space mission to be viable, the colonists must be able to harvest, process, and utilize local available resources at the destination, rather than bringing those resources from Earth.
It's the difference between packing a cooler for a weekend camping trip and moving into the wilderness permanently.
That's a great way to put it.
If you are packing a cooler, you bring all your water, all your food, all your fuel. But if you are settling, you have to find a river. Instead of shipping thousands of gallons of drinking water to Mars, ISRU dictates that we send automated rovers to mine subsurface glacial ice, melt it, and purify it.
Right.
Instead of shipping thousands of tons of liquid methane rocket fuel for the return trip, ISRU dictates we use the Sabatier reaction to synthesize methane directly from the carbon dioxide in the Martian atmosphere.
And the utilization of biological agents to convert local regolith into agricultural soil is the ultimate expression of ISRU. NASA's overarching Moon-to-Mars architecture relies completely on this principle.
Yeah, it's not a side quest.
No. Finding a way to use the ground beneath our feet on Mars is not an optional bonus or a side experiment. It is a critical, load-bearing pillar of the entire colonization effort. By leveraging these fungi, we mitigate or entirely negate the need to transport thousands of tons of Earth soil.
Which means biology is fundamentally the most efficient, cost-effective manufacturing technology in the universe.
It really is.
If you try to solve the problem with mass, you fail. If you bring a ton of Earth soil, you only ever have a ton of Earth soil. And over time, as nutrients are depleted, you eventually have less.
It's a depreciating asset.
But biology is infinitely scalable. You can launch a single lightweight test tube containing a few grams of cultivated glomeromycota and trichoderma spores. It weighs almost nothing.
Grams, literally grams.
But once you introduce it to the Martian regolith and provide a carbon source, it self-replicates. It expands exponentially. The biology builds the infrastructure for you on site using the local toxic rocks as raw material.
This scalability requires a profound psychological and logistical shift in how we conceptualize human spaceflight.
How so?
Currently, our presence in space, primarily the International Space Station, is defined by an open-loop supply chain. The astronauts on the ISS are brilliant, highly trained visitors, but structurally, they are dependent on a constant umbilical cord to Earth.
The resupply ships.
Cargo Dragons and Cygnus spacecraft regularly deliver fresh water, vacuum-sealed food, and nitrogen tanks.
Yeah.
Trash and waste are loaded onto disposable capsules and burned up in the atmosphere. It is a linear flow of resources.
But the settlers on Mars won't have that umbilical cord. When the transit window closes, they are entirely on their own for up to 2 years.
Therefore, the linear flow must become a closed circle. The settlement must operate as a strictly governed, autonomous, closed-loop life support system.
A closed loop.
In a true closed-loop system, governed by the laws of thermodynamics, matter is neither created nor destroyed. It is only transformed.
Right.
Every single atom of carbon, every molecule of nitrogen, every drop of water must be aggressively tracked, utilized, recovered, and repurposed. The concept of waste ceases to exist.
It's all just raw material for the next cycle.
Exactly. And within that ruthless, closed-loop economy, these engineered microbiomes are the engine that keeps the loop turning. They are the primary recyclers.
That transition from an open-loop visitor to a closed-loop settler is a massive paradigm shift. And we know this isn't just theoretical modeling occurring on a chalkboard in a propulsion lab.
No, it's happening right now.
We have to look at the tangible, real-world experiments happening because researchers are actively simulating these closed-loop ISRU systems, and the results they are getting are both bizarre and incredibly promising.
The empirical data coming out of experimental space agriculture is continually challenging our assumptions about biological limits. We are pushing terrestrial biology into completely novel synthetic environments and finding that the adaptability of life is far more robust than we anticipated.
A perfect example of this adaptability is the recent duckweed experiment.
Oh, the duckweed study was phenomenal.
Right. Researchers wanted to test biological scaling in a nutrient-poor environment. They took a highly engineered Martian regolith simulant, a substrate manufactured to perfectly mimic the crushed basalts, the lack of organic matter, and the specific inorganic chemistry of Mars.
Basically fake Martian dirt.
Yeah. And instead of using fungal spores, they introduced a different biological pioneer, cyanobacteria. They added just 1 single gram of cyanobacteria to the regolith, and from that microscopic foundation, they successfully cultivated and harvested 27 grams of duckweed.
A 27-fold increase in biological mass from a 1-gram initial input is a staggering return on investment.
It's huge.
Especially in a simulated extraterrestrial substrate. And the choice of organisms here is very deliberate. Let's look at cyanobacteria first. Unlike fungi, which are heterotrophs that break things down, cyanobacteria are autotrophs.
So they photosynthesize.
They photosynthesize. But crucially, many cyanobacteria are capable of nitrogen fixation. They can take inert nitrogen gas from the atmosphere and convert it into biologically usable ammonia.
Oh, wow. So while the fungi are the experts at mining the solid rock for phosphorus and iron, the cyanobacteria are the experts at pulling nitrogen out of thin air and injecting it into the sterile regolith.
They act as the vanguard, fertilizing the simulant. And then there is the choice of the crop itself. Duckweed.
Why duckweed?
Duckweed, which is part of the Limnoides subfamily, is an aquatic plant that is fundamentally optimized for rapid biomass production. It doesn't waste energy growing thick, woody stems or complex, deep root structures.
It just floats.
It simply floats, absorbs nutrients directly from the water or saturated substrate, and clones itself at an astonishing rate. It has one of the fastest doubling times of any complex plant.
That's incredibly efficient.
Furthermore, it is incredibly dense in protein and essential amino acids. In a closed-loop habitat where space and time are your most constrained resources, a rapidly multiplying high-protein crop like duckweed, powered by nitrogen-fixing cyanobacteria, is an ideal biological machine.
But the research doesn't stop at simple aquatic plants. They are pushing this into complex, familiar food crops. Other experimental teams have successfully harvested actual edible chickpeas grown in lunar regolith simulant.
Lunar chickpeas.
We are talking about the very real possibility of colonists making hummus out of simulated moon dust.
It's an amazing image.
But the methodology behind how they achieved that chickpea harvest brings us right back to the brutal reality of the closed-loop system, and it requires a strong stomach.
It does get a bit visceral here.
Tests have conclusively shown that you can vastly boost the growth rates and yields of crops like green beans and chickpeas in Moon and Mars simulants by introducing a very specific, locally sourced, highly concentrated fertilizer.
And that fertilizer is?
Human urine and recycled sewage.
It is the ultimate unfiltered realization of the settler mentality. We established earlier that in a closed-loop habitat, the concept of waste is an illusion of open system thinking. Waste is a luxury that colonists cannot afford. On Earth, we spend billions of dollars building massive infrastructure to flush our biological byproducts into the oceans or lock them in treatment plants.
We just want it to go away.
On a Mars settlement, human urine and processed solid waste are among the most valuable commodities in the entire habitat.
Let's break down the biochemistry of why that is. The Martian regolith is completely devoid of bioavailable nitrogen, phosphorus, and potassium. But human urine is incredibly dense with urea.
Very dense.
When urea breaks down, it converts into ammonia and nitrates, which is exactly the highly soluble, readily available nitrogen that a plant needs to rapidly grow foliage.
Exactly.
Solid human waste, once it is thoroughly sterilized, composted, and broken down by the engineered fungal microbiome, provides the exact organic carbon structure that the crushed regolith desperately lacks.
The human body is seamlessly integrated into the agricultural infrastructure. You are no longer just a consumer standing at the end of the supply chain. You are an active, structural component of the nutrient cycle.
You are part of the farm.
We eat the chickpeas, our bodies extract the caloric energy and specific proteins we need to function, and we excrete the remaining chemical building blocks. The engineered fungal network, the trichoderma and AMF, acts as the interface.
Processing our waste.
The fungi process our biological byproducts, combine them with the inorganic minerals they mine from the crushed space rock, and synthesize a complete nutritional profile for the next generation of crops.
It is a profound, closed-loop biochemical engine, but you really have to pause and imagine the visceral reality of being that first colonist walking into the habitat cafeteria.
Oh, man.
You've endured an 8-month transit through deep space, subjected to microgravity and radiation. You sit down at a metal table, and the hydroponics team hands you a plate of green beans and chickpeas.
Bon appétit.
Right. And, for a biological fact that those vegetables were grown in toxic, irradiated moon dust, physically glued together by microscopic alien-looking fungi, and generously, systematically fertilized by the recycled urine and processed sewage of you and your 5 crewmates.
It really takes the trendy concept of farm to table and pushes it to an absolute existential extreme.
It really does. It demands a level of ingenuity and psychological resilience to look at toxic space dust, human waste, and microscopic fungi and see a thriving, life-sustaining green bean farm.
It requires a total paradigm shift in how we view our relationship with the environments we inhabit. We are taking the harshest, most barren, unforgiving environments imaginable, places that have been dead since the birth of the solar system, and we are forcing life to take root there.
It's defiant, almost.
We aren't doing it with brute force or sheer steel. We are doing it by leveraging the deep, ancient, interconnected synergies between terrestrial plants, fungal networks, and our own mammalian biology. We are becoming the vector by which the Earth's biosphere spreads to other planets.
Let's pull the threads of this entire journey together. We started with this terrifying high-stakes scenario, standing in a pressurized habitat, staring down a pile of dead, toxic, nutrient-devoid Martian regolith.
The dirt problem.
A substrate of crushed glass and battery acid that actively resists life. We explored how solving that problem isn't a matter of synthesizing millions of gallons of liquid fertilizer, because the crushed rock lacks the biological architecture, the sponge, to hold it.
It just washes away.
And we explored how the solution isn't just loading a rocket with trillions of dollars worth of Earth soil, because the tyranny of the rocket equation makes the mass penalty impossible.
The only viable path forward relies on elegant, highly evolved, microscopic biology.
We discovered how deploying a microscopic army of beneficial fungi, specifically ancient phyla like Glomeromycota and resilient strains like Trichoderma, acts as a biological master key.
Unlocking the rock.
These organisms penetrate the microscopic pores of the inorganic rock. They excrete organic acids that metabolize toxic elements and unlock trapped minerals. They excrete glemalin to physically glue the dust together into a moisture-retaining architecture.
Rebuilding the house.
They sequester the heavy metals into their own microscopic vaults. They literally engineer a brand new habitable microbiome from the inside out.
And in executing this microscopic terraforming, they solve the massive, overarching logistical hurdle of in situ resource utilization. They allow us to truly live off the land.
It's the key to everything.
This biological integration is the mechanism that makes long-term space settlement mathematically, economically, and physically possible. It is the crucial technology that severs the permanent umbilical cord to Earth and transitions us from being transient visitors into permanent settlers of another world.
It is absolutely staggering to realize that as we look out toward the future of interstellar colonization, our greatest tool isn't a new type of fusion drive or a stronger titanium alloy. It's the exact same biological partnership that allowed the very first aquatic plants to crawl onto the barren rocks of prehistoric Earth 500 million years ago.
We're bringing the old ways to the new worlds.
We are taking the oldest evolutionary tricks in the biosphere's playbook and using them to conquer the stars.
The elegance of that biological continuity, it's profound. But as we wrap up, I want to leave you with a final, broader perspective to consider.
Oh, please do.
We spend so much intellectual energy and billions of dollars developing this highly specialized biotechnology. We are engineering microscopic fungal ecosystems capable of literally squeezing life and sustenance out of dead, toxic, irradiated space dust.
Right, for the astronauts.
But the implications of this technology don't stop at the edge of the Martian atmosphere. Consider what this exact same fungal technology, this engineered microbiome capable of thriving in heavy metals and zero organic matter, could do for the barren, degraded, or deeply polluted environments right here on our own planet.
Oh, wow. That is a staggering implication.
We have vast tracts of terrestrial land that have been functionally destroyed. We have brownfield sites soaked in industrial chemical spills, strip mine mountains stripped of all topsoil, and massive swaths of agricultural land rendered sterile by decades of over-tilling and synthetic pesticide abuse.
We have our own dead dirt problems.
Exactly. If these specific strains of beneficial fungi are hardy enough to terraform the toxic regolith of Mars, they possess the exact metabolic pathways required to heal the Earth. The bioremediation technology we are building to survive the hostile environment out there might be exactly the toolset we need to thrive and restore the biosphere down here.
That is an incredibly powerful thought to end on. The idea that by learning how to grow chickpeas in irradiated moon dust, we might accidentally unlock the exact biological mechanism needed to fix the broken soil in our own terrestrial backyard.
It's a hopeful thought.
It completely reframes the value of space exploration. It is something to deeply consider the next time you look at a patch of dirt. Keep wondering, keep questioning the mechanics of the world around you. We'll be back to explore the next frontier soon. Until then.